your Crash Course 7. So if you're in this video, I am expecting that you already finished watching Crash Course 1 to Crash Course 6. So CC7 will cover our carbohydrate metabolism. So before we start our discussion, let me give you a quick recap of what we have already discussed in Biochem. Class, remember, on our first two videos, we discussed biomolecules, emphasis on its structure, function, and properties. Oh, check. Alright, so for our third and fourth video, recall, our topic is your cellular respiration. And here on our CC7, we will cover your carbohydrate metabolism. Sir, nasan po siya dyan? So here in this box, so this will include your gluconeogenesis, glycogenesis. So these two are glycogen metabolism, your PPP or pentose phosphate pathway, and lastly, your photosynthesis. Class, recall, we are already done with our discussion on glycolysis. So kasama na po siya on our cellular respiration discussion. Diba? Here, we discussed glycolysis and we know that it's just a simple conversion of your 1 mole glucose to produce 2 moles of pyruvate. So again, this is a catabolic reaction in order to generate energy in your cytosol. Alright, and we have 3 regulated enzymes, namely hexokinase, phosphofructokinase, which is the committed enzyme in your glycolytic pathway, and lastly, we have pyruvate kinase. Remember our relevant products, stoichiometry, 1:2. 1 mole glucose produces 2 moles pyruvate, 2 moles ATP, and 2 moles NADH. Diba, sir, ano nga po ang next na process? So we know, class, that's your pyruvate metabolism. So here, we divide it into 2, diba? Anaerobic for your fermentation process, and your aerobic for your oxidative decarboxylation process. Class, all of them are catabolic. We produce energy, but here, we also use your energy in the form of NADH. So catabolic siya because we degrade your original substrate pyruvate in order to generate key products. So class, let's check their locations. Fermentation processes occur in your cytosol, while your pyruvate conversion to acetyl-CoA is inside your mitochondria. Sir, saan nga po specifically? Sa mitochondrial matrix. Class, here I will not going to discuss the key enzymes further, but I would like to highlight that your pyruvate BH again is the committed step to your citric acid cycle. Diba? For last hour, products, we have your 1 mole of pyruvate in all of your fates of pyruvate discussion. For alcohol fermentation, products 1:1. 1 ethanol, 1 carbon dioxide, and 1 NAD+. So class, I would like to clarify here, in our fermentation process, both alcohol and lactate or lactic acid, we regenerate NAD+. Diba? So here in our lactic acid again, 1 lactate ang key product, and lastly for our oxidative decarboxylation, check, 1:1 pa rin ang ratio. 1 acetyl-CoA, 1 NADH, and 1 carbon dioxide, are the products of your oxidation of 1 mole pyruvate. Diba? So let's go to our third process in cellular respiration. So class, again, this is still our quick recap to bridge our discussion to your Crash Course 7. For TCA cycle, here is just the oxidation of your fuel molecules. Class, later on in our discussion, you will see that all carbohydrates, lipids, particularly your fatty acids and amino acids, can generate acetyl-CoA. That's why they are called fuel molecules. Okay? So they can generate large amount of energy. Ano ulit yung form ng energy? That is your ATP. Okay, very good. So here, class, unique po sa citric acid cycle because it is amphibolic. Alright, catabolic, it generates energy. Anabolic role, of course, the intermediates of that cycle serve as precursors for biosynthesis of your macromolecules in our biological system. Diba, I emphasize na your anaplerotic reactions and their importance in replenishing our TCA cycle intermediates. Okay, so moving forward, location. So for citric acid cycle, of course, mitochondria na po ang location until the last process of cellular respiration. Diba, similar with glycolysis, we also have here 3 regulated enzymes, si citric synthase, si isocitrate dehydrogenase, and lastly, si alpha-ketoglutarate dehydrogenase. Class, here, I think why it's difficult to memorize the energy products of TCA cycle because ang dami. Alright, so here we have 1 mole acetyl-CoA will yield first 1 GTP. Class, remember, it's just equal to 1 mole of ATP. We also have 3 NADH, 1 FADH2, and lastly, as a byproduct, we have 2 carbon dioxide. And finally, ayan, we have your electron transport chain. Diba, recall? I think you enjoyed our discussion here. This is just a series of electron transfer, and we know that it is catabolic because, sir, bakit nga po? It generates a large amount of energy in the form of ATP. Okay, sa location, of course, guys, I have presented to you that these complexes, complex 1 to 4, including your unofficial complex 5, your ATP synthase, are embedded in your inner mitochondrial membrane. I hope that's clear. So now with our regulated enzymes, class, again, let me remind you, we have for each complexes, we have their respective inhibitors. But complex 4 and complex 5 have a lot of inhibitors. So meaning, sir, they are the 2 most highly regulated enzymes in electron transport chain. Yes, that's why we listed it here for emphasis. And lastly, with our products, class, di ba nag-compute pa tayo? We did an ATP accounting from our last video. And we know that for 1 mole of glucose, we can generate 30 to 32 ATP. O sir, bakit nga po ulit siya range? Again, it depends on the mitochondrial shuttle used by our biological system. For instance, si malic aspartate shuttle, ang kanyang net ATP yield 32, while si glycerol 3-phosphate shuttle, ayan, it can only have a maximum of 30 ATP as its net yield. And class, don't forget our byproduct H2O that is produced upon reduction of your oxygen molecule. Diba, oxygen is our final electron acceptor in your electron transport chain. Okay, so class, I hope that you are again eager, excited, and there's a thirst for knowledge here in our next discussion. So here is our carbohydrate metabolism content. And you know, class, during my time, this is actually my comfort metabolism. My taray, sir, my favorite metabolic pathway. Yes, because I think this is relatively easier to digest. Diba? Let's check the content. So first, pentose phosphate pathway. Second, glycogen metabolism. And third, we have gluconeogenesis, somehow considered as the reverse of your glycolytic pathway. But later on, you will see that it is not really, okay? So class, again, later on, you will realize kasi that these 3 pathways compared to our first set of reactions or pathways in cellular respiration, these 3 are relatively short. That's why favorite ko sila since madaling ma-familiarize yung nangyayari or what's happening in specific individual reactions of each of these pathways. Okay, so class, as an overview, here is our relevant reactions or pathways in carbohydrate metabolism. So recall recall, for carbohydrate metabolism, this involves, first one, your breakdown, okay? Catabolic reactions, storage, so meaning this is building up of your macromolecules such as your polysaccharides, so this is anabolic, diba? And conversion of glucose to meet cellular energy and biosynthetic demand. So of course, we still have the discussion of bioenergetics in this topic. O diba, ang main character natin for carbs is glucose. Remember, glucose is a monosaccharide that is on aldohexose. So let's check the processes here in your carbs metabolism. So firstly, of course, fermentation. Ay sir, parang done na tayo dyan in our discussion on fates of pyruvate. Yes, and class again, our conversion here is from glucose, pointing to your products lactate and lactic acid fermentation. And in alcohol fermentation, we have your alcohol, in particular, ethanol. Okay, ethanol usually ang end product. O second, ayan, so we already tackled your cellular respiration. Again, from your glucose, total oxidation of glucose will yield water and carbon dioxide as byproducts. And of course, class, remember, we generated here your energy. All right, and thirdly, ayan, this will be our first topic for this carbs metabolism discussion, your pentose phosphate pathway. O class, ito na yung teaser natin. You can see here, from glucose, we will convert it to ribose, a component of your nucleic acid. And again, we have a new character here, si NADPH. So later on, we will see the difference of NADPH relative to your NADH in terms of structure and their functions inside our cells. Okay, so next, ayan, si gluconeogenesis. O, di ba sir, sabi mo kanina, hindi reverse si gluconeogenesis ng glycolytic pathway. Yes po, because as you can see here, aside from your pyruvate, we can also use other substrates here in gluconeogenesis, or we abbreviate it as GNG. Or you can see lactate, amino acids, and glycerol as possible substrates of GNG in order to form glucose. O class, here, usually active si gluconeogenesis when there is a low blood glucose concentration. Alright, because again, as we have discussed, need natin i-maintain ang standard concentration ng blood glucose natin. I hope that's clear. Now, we also have your digestion or absorption. Diba, we briefly discussed your digestion as a part of carbs metabolism and cellular respiration. So, si starch, it is degraded mechanically in our mouth through digestion and absorbed in our small intestine through the use of our enzyme specific for cleaving our starch, or in general, our carbohydrates. So, we have your amylases or glycosidases. Alright, so lastly, we have your glycogen metabolism. Now, as you can see, somehow reversible po si glycogen metabolism, but they have different sets of enzymes that catalyze the specific reactions. Okay, so for glucose, in order to be converted to glycogen, that's your glycogenesis pathway. Alright, so for the reverse, from glycogen to glucose, that's your glycogenolysis. Okay, so class, when we say lysis, diba, we degrade your molecule from glycogen, we will degrade it, okay, to your glucose molecule. So I hope that's clear. Again, here is the summary of our carbohydrate metabolism with their main role and the fates of glucose. First, PPP or pentose phosphate pathway. O class, please, at this point, please also familiarize the abbreviation that we are using to your or for your metabolic pathways. O sir, ano po ang main role again ni PPP? Again, si NADPH ang main product niya. So this acts as a reducing power needed by our cell. Okay, so later we will see the specific functions ng NADPH inside our body. And secondly, again, it's important that our PPP supplies a component of nucleic acids, di ba? Ribose 5-phosphate, the sugar component ng ribonucleotides. Alright, so for the glucose phosphate, again, it's diverted for biosynthesis. That's why it's also an anabolic pathway, si PPP. And of course, redox reaction. Alright, so let's move forward to our next pathway here in carbs metabolism. That is your glycogen metabolism. O sir, ano po ang main role? Storage and mobilization ng glucose. Class, again, we have two different pathways here as we have seen in our overview slides. Glycogenesis for storage, okay? And glycogenolysis for mobilization ng glucose. So ang glucose phosphate here, of course, kapag glycogenesis, it's stored as glycogen. And kapag glycogenolysis, ayan, we release it as glucose 1P. So class, again, we are resorting to our glycogen metabolism in order to maintain our blood glucose concentration. Again, we have an optimum concentration of blood glucose. Alright, so third, ayan, si gluconeogenesis is just a glucose synthesis from non-carbohydrate substrates. O diba, from our overview slides, we have your lactate. We also have your amino acids and glycerol. And of course, activated po si gluconeogenesis during fasting in order to increase the concentration of glucose, particularly in the blood. And lastly, we have photosynthesis. O class, again, si photosynthesis is specific in plants. Recall, this is your glucose production using light energy in our plant organisms. And here, ano po ang fate ng glucose? Of course, we synthesize here your glucose from our initial substrate, carbon dioxide, and water. That's why these two are important for plant survival. O class, ready na with our first pathway for carbs metabolism? So that's your PPP. Si PPP, we also call it your phosphogluconate pathway or hexose monophosphate shunt. That's because phosphogluconate is a part or a substrate in our PPP, and of course hexose monophosphate because ang product natin, ang nature niya or ang property niya is a hexose and a monophosphate. Okay, so here we can see the overview of our pentose phosphate pathway. Class, let's discuss the overview of your pentose phosphate pathway. First, earlier I said that mainly ang metabolic type ni PPP ay anabolism. Alright, because we use PPP in order to generate other biomolecules. For example, si ribose 5-phosphate. But in reality, it is actually amphibolic. O sir, bakit po? Because again, this pathway can generate energy via your reducing equivalents. O sir, ano pong example ng reducing equivalents? Si NADPH po. Alright, so again, ang conversion natin here, glucose 6-phosphate to pentoses and reducing equivalents. So class, I think familiar na kayo kay glucose 6-phosphate. So we have encountered this substrate in your glycolytic pathway. Di ba nga, I said that's why si PFK1 ang committed step in your glycolytic pathway because si glucose 6-phosphate can funnel to other pathways such as your PPP. Okay, for relevant products, ayan. So we have two again, NADPH and ribose 5-phosphate. For location, so mainly class, particularly in mammals, your PPP occurs as well in the cytosol and it's most active in the liver, mammary gland, and adrenal cortex. So class, these sites kasi or location need a lot of reducing equivalents in order to prevent damage in our organs. Okay, so class for the phase, sir, parang similar siya ng glycolytic pathway. Yes, di ba recall, sa glycolysis we have two phases as well, si energy investment and si energy payoff phase. Here in PPP, we also have two, si oxidative and si non-oxidative. And later on, you will see that very unique po ang phases here ng pentose phosphate pathway. Para siyang switch, we can turn it off or we can turn it on depending on cellular needs. So second to the last, ang oxygen requirement ng PPP ay not that strict. Again, it's anaerobic, so it will still proceed with or without the presence of your oxygen. And finally, class, here is the overall reaction of your pentose phosphate pathway. O see, here our stoichiometry, 3 glucose 6-phosphate ang need in order to generate 1 molecule ng ribose 5-phosphate and in turn 6 moles ng NADPH. O diba? So again, 3 times tayong mag-undergo ng PPP in order to generate ribose 5-phosphate. So class, here are the phases of your PPP. So again, your oxidative phase is here in our reaction diagram. So parang ang short ng discussion here in PPP. Yes po, as I said earlier, relatively shorter ang pentose phosphate pathway natin. O check nyo, in oxidative phase, approximately we have 3 different reactions that we will tackle on the succeeding slide. So again, we have your oxidative phase. Ano ang function? Again, to generate NADPH and your substrate, your final product, ribulose 5-phosphate. And class, again, from the name itself, this is an oxidation reaction which will be convert glucose 6-phosphate to its ribulose 5-phosphate final substrate. Okay, for non-oxidative phase, ayan, we rearrange your sugar to yield your ribose 5-phosphate. So class, again, si ribose 5-phosphate is a carbohydrate. And of course, aside from R5P, we also have glycolytic intermediates as our products here in non-oxidative phase. And alam nyo ba class, these intermediates can enter as well our glycolytic pathway. Sir, kailan po? When we need energy. Because again, si glycolytic pathway can produce energy as it is a catabolic pathway. Mainly, catabolic ang function ni glycolysis or glycolytic pathway. So class, again, unique ang two phases. Sir, bakit nga po ulit? Because it can switch on or off independently depending on cellular needs for NADPH, ribose 5-phosphate, or glycolytic intermediates. So class, ready na ba with our individual reaction? So first, we will discuss the oxidative phase. So class, again, we only have 3 main reactions in oxidative phase. O sir, ang dali naman. O let's go na. Ayan. So here are our first two reaction. Actually, yung mga reactions here in your biochem, they are just repeating, diba? In different metabolic pathways, we have the hydrogenation, hydrolysis, phosphoryl transfer, and SLP, diba? So first, here in our pentose phosphate pathway, or PPP, we have the hydrogenation. And class, alam nyo ba? This is the committed step of your PPP. So si glucose 6-phosphate, again, this is from your first reaction ng glycolytic pathway. So as you can see, we will just oxidize your glucose 6-phosphate or G6P via the G6PDH or glucose 6-phosphate dehydrogenase reaction. O class, check. Here instead of NAD+, we use NADP+. So this dehydrogenation reaction initiates our pentose phosphate pathway. And later on, you will see the generated NADPH, the first NADPH, is used for reductive biosynthesis and antioxidant defense. O, diba? So here, again, we converted G6P to 6-phosphogluconolactone. O, check the structure. Again, oxidation reaction siya. And here, ang change in the structure is the reduction or the introduction of your carbonyl group in the ring of your glucose. Alright, so again, this reaction produces the first NADPH. Again, this is the regulatory step or the committed step of pentose phosphate pathway. And lastly, again, its purpose mainly is to initiate or start the oxidative phase of PPP. O, diba? Here in PPP, particularly in oxidative phase, straightforward ang reaction. Again, class, similar with glycolysis and your TCA cycle, no need to remember the structure. So just remember the essentials, diba? We always summarize the essentials. Moving forward, we have your 6-phosphogluconolactone converted to 6-phosphogluconate. As you can see here, guys, much better i-visualize ang structure ni gluconate and its Fisher projection model. O, sir, preparatory step yan, no? Yes, and here, we use our lactonase for hydrolysis. And this is the purpose, again, preparation for succeeding oxidative reactions. O, let's go to the last reaction ng oxidative phase. Hi, sir, ang dali pala. Yes, and this is again, class, another oxidation or redox reaction. O, familiar now with oxidative decarboxylation? So class, again, in this reaction, simultaneously occurring ang ating oxidation with our decarboxylation. But again here, we will use NADP+ as our coenzyme. Check natin ang reaction from 6-phosphogluconate from our previous reaction. We will convert it to our final substrate ng oxidative phase, your ribulose 5-phosphate. And here, we use our 6-phosphogluconate dehydrogenase enzyme with liberation of carbon dioxide. And this reaction completes the oxidative phase by NADPH production. So class, later, we will compare ang function ng 2 NADPH here in our oxidative phase. Again, they have different fates kasi, and of course, meron silang different functions. But before we go on that topic, this is the summary of our oxidative phase. O, diba ang reaction again, from glucose 6-phosphate, we will convert it to ribulose 5-phosphate with conversion or with production of 2 NADPH, our reducing equivalents. Alright, so class, for the process, conversion of G6P to ribulose 5-phosphate. O, sir, ba't hindi kaya natin i-convert directly to ribose 5-phosphate? Again, class, kailangan natin ng 3 rounds of PPP in order to generate your ribose 5-phosphate. Again, ang energy yield, 2 NADPH per glucose 6-phosphate consumed with your byproduct, 1 carbon dioxide. And that's it for our oxidative phase of PPP. O, class, don't be scared, huwag mo overwhelmed. This is the non-oxidative phase of your PPP. Seems intimidating at first, diba? So of course, this is just the diagram that shows how non-oxidative phase works. So of course, we tabulated it because hindi naman lahat ng intermediates are essential. As you can see, familiar na po kayo sa intermediates here in our non-oxidative phase. So some of them are glycolytic intermediates. And as we have discussed earlier, some of them are being fed again to your glycolytic pathway when cells need energy. Okay? So here is the tabulated form of our non-oxidative phase. So no need to remember their structures. So for our first two reactions, ito class is just the conversion of your ribulose 5-phosphate to 2 different sugars. O, diba? So here, specific po ang enzyme na nagkakatalyze ng each reaction. For our first preparatory reaction to your non-oxidative phase, we have the conversion of ribulose 5-phosphate to ribose. Alright, so here, our specific enzyme, isomerase po. So this is just an isomerization reaction. And the second preparatory reaction is from ribulose 5-phosphate to cellulose 5-phosphate. And here, epimerase. Okay, so class here, tip ko here is remember this by the mnemonic RICE. Diba? Ribulose, isomerase, cellulose, epimerase. Alright, so now that we already generated ribose 5-phosphate, depending on the cellular needs, of course, your non-oxidative phase is flexible. Again, it depends. The non-oxidative phase function is flexible, meaning nag-a-adjust po ang reactions depending on your cell's needs. Alright, so for our second reaction here, si ribose 5-phosphate and cellulose 5-phosphate, which are both products of our preparatory reaction, can condense in order to rearrange. Alright, so producing G3P instead of heptulose 7-phosphate with your transcriptase enzyme. So sir, ba't may with TPP yan? Of course, this is an important coenzyme of your transcriptase, your thiamine pyrophosphate. Class, remember, di ba? Ang main function po ng TPP again is to aid in transfer of your aldehyde groups. So sir, meaning merong transfer of aldehyde groups occurring in our second reaction? Yes, as well as on our last reaction here. Alright, so going back for our third reaction, from our products on the second reaction, another round of rearrangement. As you can see here, class, our second to fourth reaction, rearrangement reactions na lang po siya, depending again on cellular needs. So here on our third reaction, G3P condensed with pseudoheptulose 7P, will yield fructose 6-phosphate and erythrose 4-phosphate. So sir, lahat ba yan is a monosaccharide or sugars? Yes po. So for third reaction, instead of ketolase, we use aldolase. Alright, so class, again, here in our third reaction, we use trans-aldolase. So for our last reaction, in non-oxidative phase, we have erythrose 4-phosphate plus cellulose 5-phosphate. Class, let's see. So erythrose 4P or E4P, that's the product of the third reaction. Si cellulose 5-phosphate or X5P, that's the reactant of our second reaction. Diba? And here, another rearrangement to yield another molecule of fructose 6-phosphate and glyceraldehyde 3-phosphate with your similar enzyme, trans-ketolase with your coenzyme, TPP. Sir, bakit po may mga nakabold na intermediates on our reactions? Because class, these two molecules, ribose 5-phosphate and erythrose 4-phosphate, are the important products of non-oxidative phase. Alright, so class, actually, our second to fourth reactions can be converted to its chemical equations counterpart. So why do we need to do that? In order for us to easily visualize our final reactants and final products here in our non-oxidative phase. Clear ba, class? So now, here are the chemical equations counterpart of our second to fourth reaction. Diba? Here, our first reactant, or first chemical equation rather, is our second reaction. Our second chemical equation is from G3P plus pseudoheptulose 7-phosphate rearranged to fructose 6-phosphate plus erythrose 4-phosphate. Class, yung last chemical equation is for our last equation, reaction as well of your non-oxidative phase. Sir, yung chemical equation po ba is based on the number of carbons in the sugar? Yes. So class, let's check the first chemical equation lang. Si ribose 5-phosphate, it's a 5-carbon sugar, C5. Si cellulose 5-phosphate, again, another C5 sugar. Now, upon rearrangement, what's our product? Again, si G3P, a 3-carbon sugar, plus a 7-carbon sugar in the form of pseudoheptulose 7-phosphate. Class, the same rationalization can be applied to our last two chemical equations. That's why, here for our second chemical equation, 3-carbon sugar plus 7-carbon sugar, product, 6-carbon sugar plus 4-carbon sugar. And lastly, C4 plus C5, rearrange, ayan, product, 6-carbon sugar plus 3-carbon sugar. And with that, we can now already calculate or sum up the final reactants and final products through our cancellation method. Diba? For our first chemical equation, we know that si C3 is also used in our reactant. So used up siya. Here, si C7, used up din siya in our second chemical equation. Ano pa ang mga sugars na ginamit for the succeeding equation? We also have your C4. Diba? So I hope you still remember our chemical equation summing up strategy. Alright, so okay na ba? Now we can conclude our final reactant and product. Class, here is our final reactant and final products here in non-oxidative phase. So clearly we can see we have 5-carbon sugar rearranged to your 2,6-carbon sugar plus 1 mole of your 3-carbon sugar. So I hope that's clear again, class. It's very important that you know our final reactant and final product. So class, if we are going to convert it in specific substrates, ayan po siya. So si 5-carbon sugar, that's in the form of your ribose 5-phosphate. Sorry class, medyo sabaw na si sir. Diba? Ang ating product, of course, dalawang 6-carbon sugar in the form of fructose 6-phosphate. And a mole or a molecule of G3P. Class, take note, ang reactions natin here in non-oxidative phase ay irreversible. Sorry, again, reversible. That's why our reaction arrows are reversible. Remember, sa oxidative phase, that's irreversible. Always po siyang irreversible. So class, here again, the conversion of ribose 5-phosphate to 2 moles of fructose 6-phosphate and a mole of G3P is irreversible. So it depends what our cells need in that specific time. Alright, so I hope that's clear. Now, we sum up our products here in our pentose phosphate pathway, both for our oxidative and non-oxidative phase. Class, get ready. Again, we have 3 products. First one is from your oxidative phase, si NADPH as a form of reducing equivalence. Diba? So again, class, different po siya with our NADH in a way that in its structure, phosphorylated po siya. But again, it's not interconvertible with your NADH. Again, they have different functions in our biological system. Again, ang main function niya is to give or provide reducing power for different functions. For instance, biosynthesis and antioxidant defense. Alright, secondly and thirdly, we have your products from our non-oxidative phase. Recall class, si ribose 5-phosphate, that's our precursor for nucleic acid biosynthesis. Diba? Nakailang repeat na ako. Si ribose 5-phosphate again is a component of your ribonucleotides. And si erythrose 4-phosphate on the other hand, ayan, precursor naman siya for biosynthesis ng aromatic amino acids, which we will discuss later on in our amino acid biosynthesis. Class, just for the sake of enrichment in our discussion, Here are the structure of our three products. Again, class, si ribose 5-phosphate for nucleic acid biosynthesis. Si erythros 4-phosphate again is for amino acid biosynthesis. And again, most importantly, si NADPH has many roles in our cell. All right, so again, this is the specific role of our NADPH. Class, let's check the reactions that produce NADPH. First one is your glucose 6-phosphate dehydrogenase reaction. Again, these two NADPH have different roles inside our body. The first one is produced in order to generate your reduced form of glutathione. Plus, very important kasi na ang form ng glutathione inside the body is in its reduced form. Because again, this reduced form can inhibit the production of ROS. Also, wait lang, ano nga po ulit yung ROS? Reactive oxygen species such as your superoxide radical, hydrogen peroxide, and hydroxyl-free radicals. Alright, so again, class, here is the specific pathway of your NADPH role in your glutathione reduction. And of course, the function of your glutathione in preventing the accumulation of your reactive oxygen species. Let's check. From our oxygen, diba, this is our final electron acceptor. So upon conversion of it in your ETC via your mitochondrial respiration or exposure to ionizing radiation, sulfa drugs, herbicides, antimalarials, or divicine, of course, these are all external drugs or external factors. So kapag exposed po tayo dyan, aside from your respiration process, we can produce superoxide radical. So the superoxide radical can form hydrogen peroxide with your protons inside the body. And of course, afterwards, ayan, another radical po ulit ang mapo-form. That's why very important po si glutathione again because it is essential in maintaining the normal structure of RBCs and of course, by preventing the cellular damage by this reactive oxygen species. So class, again, familiarize yourselves with the abbreviation of reduced glutathione, GSH po. And on the other hand, your GSSG is the oxidized form of glutathione. O class, C ang ating integration ng NADPH. Again, from your reaction ng pentose phosphate pathway, your NADPH will reduce your glutathione via the glutathione reductase reaction, and this reduced glutathione will inhibit the oxidative damage to our biomolecules and most importantly, class, sa structure ng ating genetic information in the form of DNA. Alright, so upon inhibition, of course, your glutathione will be back in its original oxidized form. Here, ang ating enzyme, is your glutathione peroxidase. That's why again, important na meron tayong maintenance of your standard concentration ng NADPH inside the body. Kaya pala sir, needs natin ng pentose phosphate pathway. Yes po. Alright, for our second NADPH, this participates in reductive biosynthesis. For instance, sa lipid metabolism, particularly sa fatty acid biosynthesis, you will see that we will use this NADPH in order to form macromolecules in the form of fatty acids, sterols, and many more. Okay? So again, first NADPH for your production ng reduced glutathione to prevent ROS or reactive oxygen species accumulation. Second NADPH, again, for reductive biosynthesis. So both NADPH participate in reducing reactions. Alright, so I hope that's clear. So now, let's proceed with our next topic, the phase switching ng pentose phosphate pathway. Class, this is our second to the last slide for our discussion of PPP. So after our PPP, you can take a short break just to reflect or absorb our PPP topic or just for sanity break. So it's important kasi na ma-avoid natin ang burnout here in our discussions. Alright, so going back for phase switching again, ang important na concept that you need to remember here is that si oxidative phase, the reactions are always irreversible. And on the other hand, si non-oxidative phase, reversible po ang reactions niya as we have seen in our detailed discussion. Class, ang phase switching again, this is heavily reliant on our cellular needs. Let's check the different conditions. First, if ang cells natin need both NADPH and ribose 5-phosphate. Class, di ba recall, si NADPH, product po yan ng oxidative phase. Si ribose 5-phosphate, product ng non-oxidative phase. That's why both phases must be active kapag both are needed. So outcome of course, production of NADPH and ribose 5-phosphate. Sir, ang dali naman ng first condition. Dagdagan natin. We have our second condition, need lang po si NADPH but not your ribose 5-phosphate. That's why si oxidative phase, active po siya. Okay? How about si non-oxidative? Well, here you can see active din po siya but it is in recycling mode. So bakit siya mag-recycle? In order to produce again your glucose 6-phosphate to have another round of your oxidative phase. Class, again, here, si glucose 6P will be fully oxidized to form your NADPH and your ribose 5-phosphate will be recycled to glycolytic intermediates in order to form glucose 6P that will be fed again in your oxidative phase. So in that case, mas marami pong NADPH ang mapaproduce ng PPP. I hope that's clear again. If need lang ng NADPH, both pa rin po ay active. Now, for our third condition, si ribose 5-phosphate na lang daw po ang need and si NADPH ay hindi need ng cells. So in this case, si oxidative po ay isa-shutdown na natin. So it's inactive but our non-oxidative is active in order to produce large amounts of R5P. So here, ano ang outcome? Glycolytic intermediates will be converted to your ribose 5-phosphate. So yan na po ba yun? Class, we have a fourth condition. Ngayon, kapag si cell really needs a lot of ATP, ayan, you can see inactive pa rin si oxidative phase because again, we do not need that NADPH. As you can see here, class, ang trend here, kapag need ng cell ng NADPH, dun lang po active ang oxidative phase. Now, for our fourth condition, si non-oxidative phase lang po ang ating active here in our PPP. So particularly, the reversible or the reverse reaction. So here, your non-oxidative phase contributes to glycolytic intermediates. Sir, same lang siya ng reason with our second condition. Yes, here, we are going to convert your ribose 5-phosphate to your glycolytic intermediates so that we can generate ATP and pyruvate for your TCA and electron transport chain. Class, again, please remember the phase switching. Naging question yan from your last year board exam. Alright, so now we summarize our discussion on PPP. Again, we have two phases. Si oxidative is responsible in converting G6P to ribulose 5-phosphate. On the other hand, si NOP or si non-oxidative phase, ayan, your sugar phosphates are converted to ribose 5-phosphate or erythrose 4-phosphate depending again on your cellular needs. Primary function, oxidative for NADPH production. Non-oxidative, again, your production ng sugars, R5P and E4P. Kailan siya nag-occur? For oxidative phase, lagi po siyang active when there is a high NADPH demand. For NOP, for non-oxidative phase, ayan, it's active when there's a high ribose 5-phosphate or ATP demands. And lastly, class, remember our regulated enzymes. For our oxidative phase, I have emphasized this earlier, si glucose 6P dehydrogenase, and for non-oxidative phase, we have two, si transketolase and si transaldolase. So they are actually affected by substrate availability and coenzyme availability for your TPP. Alright, so now here is our short discussion ng regulation of your three enzymes, si G6PDH, si transketolase, si transaldolase, and I think meron pa tayong additional enzyme here. Ayan, your 6-phosphogluconate dehydrogenase. Class, let's see. For your first two enzymes, these are your enzymes in oxidative phase. So similar lang sila ng inhibitor and activator. And recall, the products of both enzymes are NADPH. Ooh, so sir, feedback inhibition again? Yes. And here again, energy rich po to, and si activator, energy deficient. For transketolase, there is no known inhibitor, but it is activated when there is a high coenzyme availability. O diba class, remember, si transketolase, it needs TPP for its aldehyde transfer function. And for transaldolase, same lang po siya with transketolase, walang known inhibitor, but its activator is dependent on substrate availability. Because again, in our non-oxidative phase, transaldolase reaction is just a rearrangement reaction. That's why it's heavily reliant sa availability ng mga initial substrates that it catalyzes. Alright, so I hope that's clear. And to end our discussion of pentose phosphate pathway, we have a did you know segment. Class, did you know that NADPH is actually very important to your cancer cells? O, Diyos ko, di ba? Your cancer's lifeline against oxidative stress. Or remember the function of NADPH in preventing the accumulation of ROS that will result to your oxidative stress. Let's see. Cancer cells experience oxidative stress from rapid growth or inflammation in its tumor environment. And ang role ng PPP here is to provide or the main source or being the main source of your NADPH in your tumor cells, especially when they are under stress from different interventions, chemotherapy, radiation, or immune cell attacks. Again, kasi ang main function ni NADPH is to provide reducing equivalence. Alright? So again, this is very important for cancer cells. So now, let's proceed with our next process, our glycogen metabolism. Class, feel free to pause the video if you want to have a short break. So now I will start the discussion again with the structure and introduction on glycogen. Diba? So remember, we already discussed glycogen in our polysaccharide discussion. Here, let's discuss it in a more detailed manner. Class, here is the essential information that you need to know about glycogen. Diba? We know that the main composition of glycogen is glucose. We know that it is a homopolysaccharide. So as you can see here, we have two different linkage type, but again, ang main linkage niya is alpha 1,4 linkages. And yung branching naman is due to the alpha 1,6 linkages. Alright, so ang storage location niya again is in your cytosol, particularly sa liver and muscle cell. And in your cytosol, ganito po siya nakapak. It's very or extensively branched. So sir, ano po ang functions in your liver and muscle ng glycogen? Class, check. Your glycogen has different functions. So for liver, it maintains blood glucose levels or we call it your glucose buffer. And on the other hand, kay muscle, it provides rapid energy for strenuous activity. Class, remember, here in our structure of glycogen, ang composition ng glucose, is about 120,000 per glycogen units. And ang branching niya is very frequent. Every 8 to 14 glucose units nag-appear ang alpha 1,6 branching linkage. Okay? So here is, ayan, a visualization of your glycogen in your liver cells. So here, class, glycogen is yung tiny violet nuts. Diba? So class, here you can see we have different or numerous glycogen storage in our liver for easy mobilization ng ating glucose. Again, it's important kasi that we maintain efficiently our blood glucose concentration. Class, with that being said, let's proceed to the overview of our two types of glycogen metabolism. So first, again, we have glycogenolysis. From the name itself, lysis of glycogen. So meaning this process is a breakdown process. So from glycogen, we will degrade it to form glucose 1P, or in the case sa liver, it's generated or its product is free glucose. Alright? So function naman again, this is important for easy source ng glucose natin, particularly when we are fasting or during physical or highly strenuous activity. Alright? So class, ang relevant products natin here, we have three. It can be G1P. So G1P can be converted to G6P and G6P can be converted to glucose. So these three relevant products are localized inside our body. So depende na po siya on our body needs. Okay? So fourth, location, always po siyang as a granule. So cytosolic granule, mainly in our liver and muscle cells. So a source of glucose for liver and a source of energy for muscle. For our energy discussion, so there's no direct ATP produced. Here, your energy is conserved as G1E. So it's mobilized when we need large amounts of ATP. And lastly, for hormonal control, yan, activated by glucagon in the liver and your epinephrine, a hormone in your muscle and liver. Class, I hope that's clear. Again, glycogenolysis, lysis of glycogen. So meaning glycogen breakdown. Let's proceed with our next pathway here in our glycogen metabolism. For glycogenesis, diba? From the name itself, birth of your glycogen. Genesis, so from glucose, we are going to synthesize glycogen. So clearly, anabolic process. For the function again, we stores your excess glucose for mobilization or for future energy needs. Again, ang relevant product natin is a polysaccharide in the form of glycogen. Diba? I repeatedly emphasize this on our discussion on polysaccharides. Glycogen is an energy storage for mammals, starch naman for plant organism. Class, check the location. Similar lang po ang location ng glycogenolysis and glycogenesis. So this is important for the availability of our substrates. Second to the last, energy use. So your glycogenesis is actually using energy. Alright? So here it requires ATP and UTP for glucose activation. And for hormonal control, it is activated by your insulin hormone. Diba recall our discussion on biologically important peptides? Nag-promise ako that we will discuss glucagon and insulin in your carbohydrate metabolism. So class, that discussion will be a part of our summary ng glycogen metabolism. So sit back and relax muna kayo. Okay? So now, let's discuss in detail the processes occurring in our glycogenolysis. Alright? So formally defined as your glycogen breakdown. So it starts with glycogen cleavage to release your glucose 1P monomeric units. So again, si glucose 1P is further converted to glucose 6P for energy use or glucose buffer in the blood. Alright, so class, similar with pentose phosphate pathway, very easy to digest po ang glycogenolysis. So here, we only have 3 individual reactions. Alright, so let's start na. First one, initial breakdown. O sir, ano po yung main reaction here? Ayan, straightforward. From your glycogen, we will cleave its monomeric units to form G1P. Sir, ano po yung monomeric units na i-cleave? So here, class, in our visualization, those are your monomeric units colored in orange. So as you can see here, we were able to liberate 8 glucose 1-phosphate monomeric units. So sir, ano po ba yung enzyme responsible here? So as you can see, phosphorylase enzyme. So in particular, it's your glycogen phosphorylase. O class, lagyan nyo na ng asterisk si glycogen phosphorylase because that is your key regulated enzyme in glycogenolysis. So it's responsible in your removal of glucose units, particularly class the terminal glucose residue. So again, the terminal glucose residue or the glucose that is readily available to it. And class, finally here in our first reaction, again, I would like to emphasize your glycogen phosphorylase removes terminal glucose residue until it reaches 4 glucose residues away from a branch point. O, diba? Let's check. 1, 2, 3, 4. That's why here are the sites of your glycogen phosphorylase reaction. And afterwards, ayan, we are now ready to proceed with the debranching of our alpha-1,6 linkage. O, plus again, required kasi na there are enough residues in our branching linkage. So again, 4 glucose residues away. For our debranching, we have a specific enzyme known as your debranching enzyme. And class, very unique ang enzyme na to because it is a single polypeptide that has 2 different function, meaning meron siyang catalytic activities on 2 different active sites. Yung first catalytic function niya is transferase activity or your alpha-1,4 transglycosylase or the glycosyl transferase activity of that enzyme. O class, here very simple, residue transfer lang po siya. And it is clearly visualized here in our picture. O class, yung transfer here na 3 glucose residues are going to be linked to your original glucose linkage. Alright? And then after that transfer, ayan, your debranching enzyme is now ready for its alpha-1,6 glucosidase activity. So that's the debranching activity of your debranching enzyme in order to cleave that remaining 1 glucose unit in your branching linkage. Alright? So again, so the branching enzyme has 2 activities, transglycosylase or transferase and your alpha-1,6 glucosidase activity. Alright, so now we arrive with our last reaction ng glycogenolysis. That is your rearrangement na lang po. Diba? As I have said earlier, your glucose 1-phosphate can be rearranged until we produce your glucose molecule in order to maintain blood glucose level. Alright, so here class, from G1P, we will convert it to glucose 1,6-bisphosphate, and from this G1,6-bisphosphate, ayan, we can now convert it to your glucose 6-phosphate catalyzed by your enzyme phosphoglucomutase. Again class, here is the reaction, G1P to G6P catalyzed by phosphoglucomutase. And again class, ang purpose ng phosphoglucomutase reaction is either to prepare our cell for glycolysis or export of your G6P to liver for future energy needs. Okay, so here class, in our arrangement reaction, we can also convert glucose 6-phosphate to your glucose with your enzyme glucose 6-phosphatase. But class, again, remember, this reaction is just localized in the liver. Again, so it occurs in liver only where free glucose is released. So sir, bakit hindi po siya pwede sa muscle? Because in the muscle, we do not have glucose 6-phosphatase. Alright, so again, it's important kasi that we saturate our glycogen or our glucose in the liver in order to maintain blood glucose concentration. Diba? So class, that's it for our glycogenolysis reactions. Let's proceed na agad with our glycogenesis. Since again, this is still under our glycogen metabolism topic. Diba? Ang glycogenesis is just the opposite of your glycogenolysis. But again, remember, we have different sets of enzyme here to avoid a futile pathway. Here, glycogenesis begins with glucose activation to form G6P and activated to UDP glucose for polymerization to form glycogen. O sir, parang ang daming intermediates needed here for glycogenesis. Of course, it's important because here we require an input of energy. O diba, recall in bioenergetics, kapag anabolic, we need energy input. O class, here is a visualization of your glycogen. O sir, ano po yung enzyme sa gitna? O class, please remember that main character enzyme. That is your glycogenin. So your glycogenin is actually a primer that signals the start of glycogenesis. So ang mechanism niya is, ayan. So it is for autocatalyzing the attachment of glucose units from UDP glucose to itself. Again class, ang monomeric units natin initially si UDP glucose. And kapag nag-attach siya kay glycogenin to its active site, ayan. So glucose na lang po ang matitira. So sir, meaning si UDP po ay source ng energy? Yes, because UDP glucose is a high energy compound, allowing your glucose to be donated to glycogen spontaneously. I hope that's clear. Again, this is a pre-glycogenesis reaction. Alright, so again, here is your glycogenin enzyme. Remember that class, again, this appeared in your board exam. I think if I'm not mistaken, 2022. O diba, meron na kayong dapat i-recall here si thermogenin from our discussion on ATC and glycogenin. Alright, so now, let's proceed with the reactions of our glycogenesis pathway. First with glucose activation. O class, this reaction is an input of energy in the form of our UTP. Class, again, si UTP kasi is suitable as source of energy here because its energy stored in its bonds are just enough for your activation ng glucose. O diba, here you can see na meron kasing preferences ang mga substrates and metabolic pathways natin for their energy currency. Alright, but again, to generalize, ang main energy currency natin ay si ATP. Okay, so here in our glucose activation, si glucose 1P, our initial substrate, will be condensed with your UTP as you can see here to generate UDP glucose. O sir, what is the reaction again? Ayan. So remember, si UDP glucose has a very high energy stored in its bonds, in its phosphate bonds particularly. O class, ang enzyme here, UDP glucose pyrophosphorylase. Alright, so class, again, the purpose is to form UDP glucose, which is the activated form of glucose. Because again, ang reason natin dyan is it is rich in high energy bonds. So again, when we say exergonic, it releases energy that is important for our next reaction in glycogenesis. So this is a series of reaction. So chain elongation yan, class. So this repeats numerously. Alright, from UDP glucose, it will be added to your initial glycogen and residues. So class, again, ang primer natin here is your glycogenine. After some time, it will form your glycogen and residues. And here, we add your glucose monomeric units as UDP glucose catalyzed by your glycogen synthase. So, sir, ano po ang product? Of course, plus 1 glucose residue lang siya. Plus, of course, your coenzyme UDP. Plus, to easily visualize the reaction, ayan lang po siya. UDP glucose will be converted or will be added to your glycogen, eliminating UDP, and this will be used again for another round of your glucose activation. Class, remember the enzyme, glycogen synthase. So here, it is also the regulated enzyme of your glycogenesis pathway. Sir, ano po ang purpose again ng second reaction? Ayan. So, of course, chain elongation, very easy to remember. So, it adds glucose to non-reducing end. Remember, class, again, ang point of attachment is alpha 1,4 po. Again, ang point of attachment is your C4, that's why it is your non-reducing end. And class, ang product glycosidic bond natin is alpha 1,4 glycosidic bond. Alright, now, with our last reaction, class C, for glycogenolysis, 3 individual reactions. For our glycogenesis, another 3 individual reactions. That's why it's so easy to remember your glycogen metabolism kasi straightforward lang ang reaction. Now, let's proceed with our branch formation. Kung meron tayong the branching enzyme, here, we have your enzyme, branching enzyme. Diba? Ang purpose niyan is to transfer a terminal segment to form a new branch. Okay, so let's check the mechanism. So first one, of course, this is the previous reaction. So si UDP glucose as our substrate, catalyzed by your enzyme, glycogen synthase, will elongate the chain until it reaches this specific elongation of the chain. Now, your branching enzyme will transfer residues or glucose residues to form branching. So class, here, it will form alpha 1,6. Okay, so again, please take note that glycogen synthase extends the branching here, ayan, to increase solubility of your glycogen, and of course, it allows your glycogen synthase to create more non-reducing ends. So class, remember kasi, ang non-reducing ends natin here, so yung point of connection sa alpha 1,4, those are non-reducing ends, they are sites of glycogen synthase, and in your glycogenolysis, your glycogen phosphorylase activity. Okay, so again, important to remember that this specific enzyme extends the branching to create more non-reducing ends, which are sites of your glycogen synthase and glycogen phosphorylase activity. So sir, mas efficient ang activity or catalysis ng synthase and phosphorylase in that case. Yes po. Okay, so again kasi, after your branching, after we transferred certain amount of monomeric units, ayan, pwede na pong mag-extend si glycogen synthase. So again, I hope that's clear. Actually, this visualization summarizes the whole glycogenesis process. So again, activation of glucose will yield UDP glucose. So si glycogen synthase will chain elongate, and afterwards, after chain elongation, ayan, si branching enzyme will transfer certain amount of glucose residues forming alpha 1,6 linkages, and yan, continue na po ulit working si glycogen synthase in extending both non-reducing ends. So here it is, guys. This one is a non-reducing end, and this one is another non-reducing end, followed by more branching. That's why napaka-efficient in terms of energy storage si glycogen. O class, ready na tayo with our summary? Ayan, to summarize your glycogen metabolism, these are the important information that you need to know. First, with glycogenolysis, again, our process is breakdown from glycogen to your glucose monomeric units. And again, active po siya when we need a lot of glucose concentration. So that's when you are fasting or when you are exercising or you're under stress. Ngayon ba you're under stress? So meaning, activated si glycogenolysis. Diba? So for tissue involvement, ayan, so bida pa rin po si liver and muscle. So class, again, in glycogen metabolism, si liver and muscle po kasi ang merong certain amount of glycogen granules. That's why, of course, sila lang ang main characters for our glycogen metab discussion. And lastly, I emphasized this earlier, for glycogenolysis, phosphorylase po ang regulated enzyme. On the other hand, ayan, again, glycogenesis is just the opposite or the reverse of glycogenolysis, in terms of the process occurring here. From our glucose monomeric units, in order to store energy, we need to build glycogen. Sir, kailan nga po siya active? Ayan, pagkakain or after meals when we have high concentration of glucose in the blood. So, kailangan nating immobilize yung blood glucose. That's why we need to synthesize glycogen. Alright, so tissue involvement, same lang po with glycogenolysis. And lastly, regulated enzyme, glycogen synthase. So class, lastly here on our summary, we need also to cover regulation discussion. Now class, don't be scared. Dalawang enzyme lang ang regulated here in glycogen method. So sir, dalawa din lang po yung enzymes for regulation. Yes. And class, as you can see here, majority ng mga effector molecules, for instance, ng inhibitor and activator are hormones. For phosphorylase, we have insulin. So remember class, the insulin is a hormone that we discussed in our biologically important peptide. So later on in our discussion, we will see its specific mechanism. Secondly, si ATP, again class, this inhibits phosphorylase reaction because there's no need to mobilize glucose for energy generation. Okay, kasi nga, we have high concentration ng ATP. And lastly, glucose 6-phosphate recall is a product of rearrangement of your glucose 1-phosphate. And glucose 1-phosphate is the product of your glycogen phosphorylase reaction. So somehow, this operates through feedback inhibition mechanism. And for activator, we have these two hormones, glucagon and epinephrine. Class, for epinephrine, this is through phosphorylation regulation. Alright? So it phosphorylates and dephosphorylates your enzymes. And lastly, we have glycogen synthase. Again, kung activator, si glucagon and epinephrine with your phosphorylase, here, they inhibit glycogen synthase. And for activator, ayan, you can see, nag-switch lang sila. Yung inhibitor ng phosphorylase, activator naman siya ng glycogen synthase. In particular, si insulin and si glucose 6-phosphate. Alright, so class, I hope that our discussion on glycogen method is clear. So again, we simplified the discussion to be board exam relevant na lang, okay? This just contains all board exam relevant information. And to end again our discussion, ayan, we have a did you know segment. So class, did you know that rarely po tinatouch ng brain natin ang glycogen? So again, our brain rarely touches glycogen until it reaches the panic mode. O, di ba? When we are panicking, ayan, we start to mobilize our glycogen. For instance, kapag stress tayo or when we are studying. Class, particularly astrocytes or those cells that store glycogen and only release glucose during extreme demand like hypoglycemia. So for instance, mental exertion. So during your studying session like this one, doing hard problems like your example problems in Phychem, Anachem, and in inorganic chemistry. Okay? So your mental exertion has been shown to lower glycogen levels. So we mobilize your glucose through glycogen breakdown. And again, class, this is occurring in specific brain regions, such as or in particular in your prefrontal cortex. So class, here's the structure of your astrocytes. So no need to memorize the structure and your terminologies here. Again, this is just a supplementary discussion in order to enrich our discussion. And class, here is, ayan. So this one, it also appears in the board exam. So there are certain glycogen storage diseases. So we have 6, all right? This is categorized as type 1 to type 6. So first one, we have 1-Geert disease, which is the deficiency in G6P enzyme. Okay, your glucose 6-phosphatase enzyme. Type 2, we have your Pompe or your lysosomal alpha-1 for glycosidase deficiency. So class, remember, these enzymes are all in our glycogen method discussion. Okay, so going back, our type 3, Chiari disease is the lack of the branching enzyme in our liver and muscle cells. Si Anderson naman is the opposite. So meron tayong debranching, meron tayong branching enzyme. So same lang siya na nasa liver and muscle. And for our top 5, ayan, this is a localized enzyme. Your McCarthy disease is the deficiency in your muscle. So muscle glycogen phosphorylase. And lastly, si ERS is your hepatic glycogen phosphorylase deficiency. Sir, baka may mnemonics ka dyan? Yes po. So class, here is our mnemonics. So, diba? Very Pinoy. Chismis ang main hobby. So first one, again, we have your 1-Geert, Pompe, we have Corey, we have your Anderson, we have your McCarty, and ERS. All right, so now let's proceed with our sample problem. So class, check your manuals. The answer is already given in your biochem notes. So here is our sample problem for glycogen method. So during a period of high blood glucose, which glycogen metabolic process is most active in the liver? O class, remember ha, high blood glucose. So ano po ang active? Is it glycogenolysis or glycogenesis? O class, I presented it earlier, di ba? So your glycogenesis is actually the one that is active during high blood glucose concentration. O very easy because again, kapag meron tayong high blood glucose concentration, we need to deplete the concentration of your glucose. So how do we do that? Of course, we need to synthesize glycogen. And in that way, we maintain the standard or the optimum concentration of your blood glucose. So again, class, to summarize, glycogenesis is the process of storing excess glucose as glycogen in the liver. So in particular, sa liver lang po siya when there is a high blood glucose concentration. All right, so class, we are now done with our glycogen metabolism discussion. Or similar with TPP, you can now take a short 2-minute break before you proceed with our next topic, which is your gluconeogenesis or GNG. All right, so now we proceed with our gluconeogenesis pathway. Class, our discussion here will be much shorter because again, almost similar lang yung certain reactions with our glycolytic pathway. O, diba? Essentially, this is the reverse of glycolysis nga. But remember, there are reactions that bypasses the irreversible steps with alternate enzyme. So class, important that there are different sets of enzymes here. Again, same reason, to avoid a futile reaction or an inefficient reaction. So here is the overview of our gluconeogenesis. So class, again, ang process, anabolic siya, sir? Yes, because we are synthesizing glucose from non-carbohydrate molecules. So class, did you know that the human brain actually consumes over 120 grams of glucose each day? That's why there are instances na lacking tayo ng glucose concentration. We need to synthesize it from non-carbohydrate precursors. Okay? So again, your glucose is made in the liver and transported to the brain in the bloodstream. So sir, how about the energy use? Yes, we have an energy use here in gluconeogenesis. Actually, class, si GNG is energetically expensive. Okay? So it consumes a large amount of ATP and your electron carrier, NADH. And in terms of location, ayan. So it is located in your liver cytosol and mitochondria and in our body part, kidney. And of course, class, our bypass enzymes, you need to memorize this. So these are the enzymes that are alternative to your irreversible steps ng glycolysis. O diba, remember, si hexokinase, si PFP1, and si pyruvate kinase. Ang first bypass enzymes natin, your pyruvate carboxylase and pep carboxykinase are alternative enzyme ng pyruvate kinase natin. Secondly, your fructose 1,6-bisphosphatase is the alternative enzyme of your PFP1. And lastly, si glucose 6-phosphatase, ayan, alternative enzyme ni hexokinase in your glycolytic pathway. O sir, wait lang, I have a question. Is GNG aerobic? So actually, class, si gluconeogenesis is indirectly aerobic. Okay? So while it doesn't use oxygen directly in its chemical reaction or chemical steps, it requires ATP and NADH. Okay? So class, remember these two energy products, they are mainly produced in our aerobic pathways, your cellular respiration, particularly class sa oxidative phosphorylation. So class, to answer your question, yes, it's functionally aerobic because it depends on energy and producing power in the form of ATP and NADH respectively, which are harnessed from oxygen-dependent processes. Okay? But partly no then because it's not directly aerobic. Okay? In the way that oxidative phosphorylation is not in its core enzymatic reaction. Alright, so I hope that's clear. Again, our possible substrates, our non-carbohydrate precursors can be lactate, alpha-ketoglutarate, and glycerol. And of course, class, you're familiar with pyruvate, the product of glycolysis. And here is the overall reaction of our GNG using pyruvate. Again, no need to memorize the overall reaction. Class, again, please remember, gluconeogenesis or GNG is not just a simple reverse of glycolysis. Okay, so now let's proceed with our overview. Okay, additional overview ng gluconeogenesis pathway. So here again is the comparison of your glycolysis and gluconeogenesis. Class, this is available in your biochem notes. Class, again, as we have discussed earlier, here in gluconeogenesis, we have 4 bypass reactions. So we tabulated it. Again, for glycolytic reactions, we have 3 reactions being bypassed by gluconeogenesis. O sir, ba't hindi equal? Because again, ang first bypass reaction is equivalent to 2 bypass reactions in your gluconeogenesis. Class, here it is. Ayan. First enzyme, pyruvate carboxylase, is the conversion of pyruvate to oxaloacetate. But class, here you can see si pyruvate kinase is the conversion of PEP to pyruvate. Class, here, ang product natin is oxaloacetate. That's why we need another enzyme to convert oxaloacetate to your PEP in order to reverse your pyruvate kinase reaction. So here, we use PEP carboxy kinase enzyme. Class, I hope that's clear. Again, for pyruvate kinase reaction, ang bypass reaction natin ay equivalent to 2 enzymes. Secondly, si PFK1, again, we know that it catalyzes the reaction fructose 6-phosphate to fructose 1,6 bisphosphate. For our gluconeogenesis, our bypass enzyme is fructose 1,6 bisphosphatase, catalyzing this reverse reaction from F1,6 bisphosphate to fructose 6-phosphate. Okay, so class, in our pathway, ayan, it is located here in front of our glycolytic reaction, but farther to our start of gluconeogenic pathway. And lastly, si hexokinase, of course, is bypassed by our G6-phosphatase or G6P enzyme. And of course, here, ang main reaction for glucose 6-phosphatase, reverse lang po ng hexokinase reaction from G6P to glucose. Alright, so here it is. Ayan. So these bypass steps are actually critical as the original glycolytic enzymes are thermodynamically. So in terms of bioenergetics, they are irreversible under cellular conditions. Alright, so class, our next discussions will be the detailed discussions on your bypass reaction. So let's start with the bypass one, which involves 2 different enzymes. So for our first reaction, this is the conversion of pyruvate to oxaloacetate with the use of pyruvate carboxylase. Class, again, I would like to reiterate, no need to memorize the structure. Here, ang purpose po ng first reaction is to consume ATP. Oh, sir, that's why it's energetically expensive. Yes. So again, it consumes ATP and uses biotin as its coenzyme, and this is the initiation reaction of your bypass reactions in GNG, particularly ng pyruvate kinase, okay, or pyruvate kinase. Sir, where is it located? Class, your first bypass reaction is in your mitochondrial matrix, all right? For our second reaction ng bypass 1, we have your conversion ni oxaloacetate to your phosphoenol pyruvate catalyzed by the enzyme PEP carboxy kinase. Class, again, here it consumes another energy in form of ATP or GTP depending on the isozyme. Okay, so this completes the first bypass reaction for your pyruvate kinase. So as you can see here, class, this is a decrease in carbon chain of your OAA to form PEP. So class, simultaneously, we also liberate carbon dioxide. Oh, you can see class here that for the first bypass, we already used 2 equivalents of ATP. That's why, again, energetically expensive po ang synthesis ng glucose from non-carbohydrate precursors. Alright, so now we proceed with our alternative pathways with our bypass 1. Class, before I forget pala, ang location ng second reaction ay nasa cytosol na. Okay? So we have a specific transport protein, particularly for your OAA. Because again, ang conversion ni OAA to PEP is occurring in your cytosol. Alright, so class, we will summarize naman the locations of our different metabolic pathways. But now again, we will also discuss your different or alternative pathways for bypass 1. So here you can see pyruvate can also be converted to PEP through your malate intermediate depending on the cell's needs and location. Okay, so sometimes kasi class, ayan, we still need to funnel your bypass reactions in different pathways in order for compartmentalization to be efficient. Again, ang first reaction kasi ng bypass 1 natin, your conversion ng pyruvate, okay, to oxaloacetate occurs in the mitochondria. And again, very busy organelle si mitochondria. That's why we have alternative pathways. So first one, from pyruvate, it will enter your mitochondria and this will be converted to oxaloacetate. So si OAA will be converted to malate. So class, reverse reaction ng last reaction ng TCA cycle. Okay? So we have a malate aspartate transport protein, and of course, your malate can be transferred to your cytosol and this will be converted back to your oxaloacetate, and tada, we converted it to PEP. Alright, so of course, similar reaction lang here, but here we started with lactate. So this is the case for your fermentation or anaerobic organisms. Okay, so this is again just an additional reaction in our alternative pathway discussion for bypass 1. So I hope that's clear. Now, we will proceed with our 2 bypass reaction. So pretty straightforward na lang yung 2 bypass reaction. For bypass 2, again, reverse lang siya ng conversion of your PFK1. Here, ang enzyme natin is yung fructose 1,6 bisphosphatase. So this is a hydrolysis reaction and its purpose is to bypass the PFK1 without ATP consumption. And location class, remember, your glycolytic pathway ay nasa cytosol, hence this reaction will also be found in your cytosol. Alright, for bypass 3, we have your G6-phosphatase enzyme. So again class, similar with bypass 2, reverse reaction lang siya ng hexokinase reaction. So here, from G6P, we convert it to glucose via the glucose 6-phosphatase reaction. And ang purpose, ayan, this is to enable glucose export from liver. And class, very unique ang location ng bypass 3. It's in your endoplasmic reticulum membrane. Okay? So class, again, si bypass 3 is actually not necessary sometimes. So diba, in our glycolytic pathway discussion, ang first reaction, ang hexokinase reaction, is just to trap your glucose inside the cell. So kapag hindi need lumabas ni glucose, of course, it will be retained as glucose 6-phosphate. That's why sometimes your bypass 3 is not necessary. Alright, so class, we have a special discussion in our gluconeogenesis. So your GNG inhibition by alcohols. Alright? So why are you weak and hungry after drinking? Class, recall our reactions in our ethanol fermentation. Diba, this is just the reverse of our ethanol fermentation. From ethanol through alcohol dehydrogenase, we will produce NADH, and this will be converted si ethanol to acetaldehyde. And consequently, si acetaldehyde, of course, this will be converted to acetate again with another round of NADH through your enzyme aldehyde dehydrogenase. Alright, so when a person like you, diba, aminin, so when you consume alcoholic beverages, the liver actually prioritizes breaking down ethanol through this reaction. So this process generates high amount of NADH, and did you know that this disrupts the balance of NAD plus NADH ratio in the liver? In particular, ayan, alcohol inhibits GNG by diverting NAD plus to ethanol metabolism. Alright, so in particular, NADH promotes reduction of oxaloacetate back to malate, and hence, ano po ang result, sir? Ayan, we do not have enough concentration ng OAA for gluconeogenesis. That's why we are weak and hungry after drinking. Tinitrick tayo ng brain natin that we are actually full when we are drinking alcohol. Alright, so one of the key consequences here again is the inhibition of gluconeogenesis. So here is the summary. Ang ethanol metabolism natin, here, ang effect niya with our NADH production, increase. Okay? So when we have enough NADH inside our cell, ang result niya, si pyruvate, i-convert to lactate because again, we have a large influx of NADH and on the other hand, si oxaloacetate will be converted back to malate. Okay? So of course, in terms of high NADH levels, ayan, so of course, depleted po ang GNG intermediates. So anong GNG intermediates? Remember, oxaloacetate po ay intermediate ng gluconeogenesis. And when we convert it back to malate, ayan, magdi-decrease ang concentration ni GNG. That's why inhibited po ang gluconeogenesis. So what is the effect? Ayan, ang overall result, hypoglycemia, meaning lower blood glucose production in our body. Okay, so class, I hope that's clear, diba? That's why some of the elderlies advise us to eat first before drinking alcohol. Para hindi tayo matrick ng brain natin that we are actually full, but in reality, we are just really have a high amount of NADH in the body. Okay, so class, another related topic here is your Cori cycle. So again, si Cori cycle is very crucial in lactate recycling. Okay, so again, this is very important in order for your lactate from muscles to be delivered back to glucose in the liver. So actually guys, your Cori cycle is the linking pathway of your glycolysis and gluconeogenesis. Check. Class, so let's start and briefly analyze your Cori cycle. Starting with our skeletal muscle where lactate fermentation occurs again. And emphasize ko yan during our discussion on fates of pyruvate. So here, doing glycolysis will convert glucose to pyruvate and with your lactate dehydrogenase, we will produce lactate in the muscles. Diba? It's very important kasi that we mobilize or we transport your lactate going back to the liver in order to prevent cramps due to lactate buildup or lactic acid accumulation. So again, yung accumulation ng lactate and proton ions will lower pH here in your skeletal muscle and thus will result into cramps. So that's the reason we need to mobilize your lactate all the way going to the liver. So in the liver, ayan, the reverse process will occur from lactate to glucose. And here, another round of your glucose conversion in the muscle will occur. And that is your Cori cycle. And class, did you know that Cori cycle actually helps maintain blood sugar during exercise or fasting? Diba? So here, when we have hypoglycemia or we have low blood glucose concentration, ayan, so your Cori cycle is on and hence it will deliver lactate to the liver in order to convert it back to glucose. And when we have normal glucose concentration, your Cori cycle is off. Again, it can be turned on or off depending on cellular needs. Again, it's very important to say that we maintain homeostasis. Alright, so to summarize our gluconeogenesis pathway, ito class, ang important information that you need to know. So again, let's reiterate, process, non-carbohydrate precursors are converted to glucose. Okay, so when it occurs, again, during fasting, prolonged exercise, or low carb intake, which will result to your low blood glucose concentration. And for our substrates, again, we have several substrates. For instance, si lactate from our Cori cycle, si alanine from our Cahill or glucose alanine cycle. So class, this will be discussed on amino acid metabolism. And lastly, glycerol from fat breakdown. So this will be discussed in detail in lipid metabolism. And for our energy use, class, check, it requires 6 high energy phosphate bonds per glucose. That's why very energetically expensive ang gluconeogenesis pathway. Alright, so for the link to other pathways, we have beta oxidation, TCA, and Cori cycle. Diba si TCA is with your oxaloacetate intermediate. Si Cori cycle is with your lactate substrate as our initial substrate. And of course, the beta oxidation, we will see it on our discussion on lipid metabolism. Class, carry pa ba? So we just have a few slides left and I will wrap up this video that covers your carbohydrate metabolism. So we will continue our discussion on the next video with our discussion on photosynthesis. Class, kapit lang, this is our regulation discussion. So similar with other metabolic pathways, it's important that you're knowledgeable with the inhibitors and specific activators of each regulated enzymes. Class, let's recall that your gluconeogenic pathway, is energetically expensive. Okay? That's why kapag low in energy ang cell, it will inhibit gluconeogenesis. Let's check. For pyruvate carboxylase, PepCK, and F16-bisphosphatase, these inhibitors are all energy deficient. ADP for pyruvate carboxylase, ADP and NADH an electron carrier for pep carboxykinase, and fructose 2,6-bisphosphate and AMP or fructose 1,6-bisphosphatase, they are all deficient in energy. Because again, when your cells are deficient in energy, it is unfavorable to do gluconeogenesis kasi nga energetically expensive po siya. So of course, the glucose production will come from other alternative metabolic pathway na hindi energetically expensive. On the other hand, with our activators, these are energy-rich molecules. So class, we can have a general trend here, diba? For catabolic reactions such as glycolysis, ang inhibitor, energy-rich. Ang activator, energy-deficient. For anabolic reaction, the opposite is true. For inhibitors, energy-deficient sila dapat, and for our activators, energy-rich. Diba? Acetyl-CoA is being fed to TCA cycle, NADH is being fed to electron transport chain. Both will be converted to ATP in your electron transport chain. So citrate is a part of your TCA cycle, and of course, we know TCA cycle is the main or the central hub of energy-yielding pathway in our cellular respiration. Alright, so I hope that's clear. So that's it for our discussion on gluconeogenesis. So the remaining discussion are supplementary and of course important for you to know. So here is our meron pala tayong did you know segment before we discuss glucagon and insulin. Class, did you know that, hibernating animals actually rely on gluconeogenesis to survive winter? So actually, class, if I recall it correctly, we call it your slow burn biochem when we are using gluconeogenesis as a means of survival. So for instance, here in our hibernating animals, as they fast for months, your hibernating animals, for instance, bears and ground squirrels, they keep just enough glucose flowing for the brain and red blood cells. So instead of relying on food sugars as their basic energy source, the animal's liver and kidneys produce sugar endogenously through your gluconeogenesis using amino acids and other body reserves such as your lactate and your glycerol. Alright, so class, let's proceed with our discussion on glucagon and insulin, the two most important hormones in carbs metabolism. So here are the information that you need to know. to know. So for glucagon, it is secreted by pancreatic alpha cells of islet of Langerhans. So islet of Langerhans are actually groups of pancreatic cells that are responsible in secreting your insulin and glucagon. So again, si glucagon is responsible in increasing blood glucose during fasting. Si insulin on the other hand, ayan, kung si glucagon alpha cells, siya naman, ang kanyang secretion mode is through its beta cells of islet of Langerhans. And of course, ang kanyang role is to decrease naman your blood glucose by enhancing glucose uptake. That's why, di ba, for diabetic patients or those who have high blood glucose concentration, of course, they need to have an injection of insulin in order to decrease or to mobilize your blood glucose concentration. Plus, here are the components of your pancreas. Sir, nasan po dyan si alpha and beta cell? Let's see. Si beta cell here is depicted in yellow. So those are your beta cell in your pancreatic cell, particularly in your islet of Langerhans. Si alpha cell, ayan, it secretes glucagon again, and here it's colored in red. So there is no conclusion naman in terms of the size of your beta and alpha cells. So it varies. And of course, class, again, the source of your glucagon and insulin appeared already in your board exam. That's why I made a mnemonic. Sir, pa-share naman. Kasi friend ko si Ariana Grande, college friend ko siya, and of course, personalize yung mnemonic. Charot. So here is the mnemonic. Ariana Grande, bad influence. Alpha cell secretes glucagon, beta cell secretes influence. So again, AGB, Ariana Grande, bad influence. All right, so class, here is the specific mechanism of your glucagon and insulin. Class, here, let's briefly discuss the enzymes participating in your gluconeogenic and glycolytic pathway and their relationship with your glucagon and insulin hormones. Class, please don't forget ha, si glucagon again, it stimulates glucose production and glycogen breakdown. In particular, sa insulin, ayan, it stimulates glucose uptake naman siya. And of course, your glycogen formation. Or here are the 4 enzymes that are relevant in our discussion. 2 of them are from gluconeogenesis, the F16 bisphosphatase and CPEP-CK or pepcarbokinase, carboxykinase, sorry. And the 2 of them also is from or are from glycolysis, PFK1 and pyruvate kinase. So let's check. In the presence of your glucagon, si F16 bisphosphatase activated because again, glucagon favors glucose production. All right, so insulin, of course, it inhibits your fructose 1,6 bisphosphatase because again, we need that glucose uptake. We do not need to produce glucose. All right, so PFK1, remember class, this is the enzyme that is bypassed by your fructose 1,6 bisphosphatase. That's why, ayan, nagkaroon lang tayo ng reversal ng inhibition and activation. So si glucagon, i-inhibit niya ang glucose uptake via your PFK1. And si insulin, ayan, it will activate PFK1 because again, this is towards glucose uptake. And for our third enzyme, similar lang po siya with your glycolytic enzyme. Sir, is that the trend? Yes po. So again, for your regulated glycolytic enzyme, lagi pong ini-inhibit ni glucagon ang kanilang activity, while for insulin, ayan, they activate it for increased glucose uptake. And lastly, for our PepCK, similar with our fructose 1,6 bisphosphatase, ang expression niya ay increased in the presence of glucagon, and the opposite is true when we have insulin hormone present. All right, for pathway shift, ayan. So this is the summary. Again, for glycolysis, your glucagon inhibits glycolysis and favors gluconeogenesis. And for insulin, it favors glycolysis and it inhibits gluconeogenesis. So class, I hope that's clear. Again, glucagon and insulin, we have Ariana Grande bad influence, diba? So alpha cells, glucagon, beta cells, insulin. And class, here is the mnemonic for our gluconeogenic substrate. Diba? We are just going to reuse the mnemonics from our glycolytic pathway. But here, we have an additional oxaloacetate. Class, get ready. Ayan. So similar mnemonic. Gosh, girl. Fast and furious now, girl. But promise, puro pepa. Ayan. What is the sound of the pig? Point. Puro pepa. Point pig. Class, again, feel free to use our mnemonics. So later on, we will summarize all of the mnemonics that I am sharing to the class. And to end our video here, I have prepared a quick quest. Class, let's see our quick quest for this crash course. So this is just a simple true or false. Let's start. Class, about PPP or pentose phosphate pathway. The PPP produces NADH. Diba? Emphasis on NADH. Is this true? Of course, this is obviously false. Again, class, NADPH po. The phosphorylated form of your NADH or your NADPH is produced here in PPP. So this is used for biosynthesis and antioxidant defense as we discussed on our discussion on PPP. All right, so next, lactate and amino acids can serve as substrates for GNG. Class, recall, for GNG again, our non-carbohydrate precursors can include lactate and amino acids. Sir, alam ko to yan. Yes po. So class, again, aside from these two, we also have glycerol and pyruvate for your GNG pathway. All right, thirdly, or our second to the last, quick quest. Glycogen is stored in kidney and muscle. Class, I have a question mark on kidney. Again, it's not kidney. Diba, remember, for your glycogen metab, it always occur in your muscle and liver. That's why this statement is false. And lastly, ayan, so PPP occurs in the mitochondria. Nako, sir, that's obviously false, diba? Because again, PPP is in your cytosol. All right, so class, I will cut the video at this point and we will continue our discussion on our carbs metabolism with photosynthesis on our next video. So class, again, thank you for listening. So take a break, and of course, to avoid burnout, do your hobbies or any activities that will make you happy. Okay, so class, bye for now. See you on the next video. Bye. Hi class! So again, as I have said from our last video, this is the continuation video on our discussion on carbohydrate metabolism. Okay? So here in this video, ang focus natin is your photosynthesis. So class, I think you are well familiar with this pathway since high school, diba? But here we are going to discuss it in detail. So as an introduction, let's watch this short animation on photosynthesis.